Welcome to Climbing the Immune Ladder, Steps for Success in Science, iiSIAR podcast, the show where we explore inspiring experiences in science and the untold stories shaping our community. Today we'll be your hosts, Janine Lückgen, Senior Postdoc in Immune-Mediated Inflammatory Diseases, and Salma Rizo, Postdoctoral Fellow at the University of Montreal, Canada. In each episode, we will dive deep into conversations with experts on innate immunity and sterile inflammation, autoimmunity, and their resolution, to bring you insights, ideas, and inspiration you won't hear anywhere else. Whether you're tuning in during your morning coffee or while you're doing your lab bench work, get ready to learn, laugh, and see the world from a new perspective. Let's get started. Today's expert is Tal Burstyn-Cohen, Professor of Development, Biology, and Cancer Research at the Hebrew University in Jerusalem. Thanks for joining us and welcome to our podcast, Tali.
Hi, so it's a pleasure to be here in this podcast. And I am a scientist at the Hebrew University in Jerusalem, Israel. And my career has been many places. I assume some questions maybe later on would touch on those things. But nowadays, I am becoming more and more of an immunologist, and hence, the relation to this podcast.
Okay, very nice. Perhaps we could start by what actually sparked your interest in immunology and specifically in innate immunity, inflammation, and autoimmunity.
Okay, well, at the beginning of my career, I was not an immunologist at all. When I was at undergraduate, I had an amazing, wonderful professor who taught me basic immunology, but I was drawn away to neurobiology, in fact, and developmental neurobiology. That's where I did my master's and PhD, and even my first postdoc. And then I transitioned into a field. It was still in neurobiology, but in the adult. And it kind of, immunology was there in the background. The lab at the time was becoming an immunology lab. And I realized that the genes and the pathway that I explore, and I would go a little bit more into that later on, is also related to immunology. And I realized that immunology and immune cells are everywhere and they affect many tissues and many organs in the body. And that's how my interest in immunology was born.
A very nice story. So, are you still working with neurobiology or are you now fully focusing on immunology?
So I work in neurobiology, but on the immune cells of the nervous system, microglia. Of the central nervous system. We work on microglia, but not only, we also work on macrophages. And I am fascinated by this interaction between immune cells and the tissues and how they maintain homeostasis, how they contribute to development, what goes wrong in diseases. I mean, I think they're major players in biology.
Yeah, absolutely. Very fascinating. Perhaps you could dig a little deeper into what findings concerning your research on microglia were most impactful or perhaps also most surprising. Is there anything that comes to mind?
Well, in particular, into microglia, these are things that are not yet published. So maybe we'll wait a little bit with that. But a surprising finding was, and maybe this is where I can elaborate a little bit about the pathway that I study, because I think it's essential already to introduce it because it's everywhere. So I study the TAM signaling. This is signaling through a family of receptor tyrosine kinases, Tyro 3, Axel and Mer, and the acronym is TAM. And when I joined my second postdoc in the US at the Salk Institute with Professor Greg Lemke, at the time there was a big controversy as to how these receptors function, who are their ligands. There were two ligands at the time, and one of them was completely there was a big debate in the field whether this molecule was really a functional ligand or not. This molecule is called protein S, and at the time it was known as a very, very potent blood anticoagulant. Has nothing to do with signaling or any cellular functions. And there were several papers actually canceling the role of protein S as a ligand and refuting one paper that identified it as a ligand. But in the lab, we had some hints as to, we thought that probably protein S is a ligand. So I had to create a tool to ask the question. I generated a conditional knockout mouse. And if you asked about challenges or, stuff, I did not know too much about molecular biology at the time. And generating conditional knockout mouse had to do with a lot of molecular biology. I just dived into it, and I got a lot of support and help from the lab and from my peers and colleagues. And at the end, wow, there was a mouse. There was a mouse, and that was a challenge for me to generate something. I knew that I want to do this. I'm learning throughout the process. Of course, you never know whether this would be informative tool to study or not. Would it be successful or not? It turned out that it was successful, and it was a big, big challenge, and it even opened up a lot of the research that I still conduct in my lab. And today we study the role of this molecule, protein S, not as a blood anticoagulant, but more in its non-canonical functions, if you like, as a signaling molecule and activating numerous and diverse cellular functions.
Wow, very inspiring. Never give up and just and overcome all the obstacles. I understand that there is a high risk of preparing a mouse model. A lot of time and effort is going into this. Was there also a time where you had a challenge that you could not overcome? And how did you deal with this?
Well, at the beginning, we had to decide where to clone our LOXP sites, and the first time just didn't work. I couldn't get it, so I just decided to move a little bit. I remember it was like moving 700 base pairs downstream or upstream, and then it worked. And there were many, many challenges because it was my first for many things. But I learned to adopt these challenges and learn from them. I remember there were tough times when I came home after a day and I was, kind of maybe even depressed, or we all know that science isn't easy. Many things don't work, but if we learn what doesn't work, then it can guide us and change, and then we see what works. At the end, we find what works, and I think that that was a big lesson for me through, and I still carry this till today. To try and to not give up, because at the end, I think it's worth it.
Yes, absolutely. Now let's talk a little bit more about your career. And I was wondering whether there were any career opportunities that you turned down and later reconsidered, or you didn't regret. Can you tell me something about this?
Um, well, I don't really think that there were any career opportunities that I didn't take. I think I see this as kind of a journey, and there are always several opportunities. You know, you take one, by default, you don't take the other one, right? And everything is flexible. So, I mean, we do what's good for us at the end, what's exciting for us. And if it won't be this, it would be something else. And science, I think, and the academia also have a lot of flexibility, and they allow us to this freedom within. So if I don't, when you mean career opportunity, do you mean like a job, or do you mean like a project in the lab? It can be anything, right? So yeah. So for a job, um, I mean, at a certain point, I had several options, and I chose one thing. I mean, nothing is forever. Um, that's my motto.
What skills do you think are most important when pursuing a career in science? I mean, outside of the scientific experience. So, what kind of soft skills do you think everyone should acquire in order to be successful?
I think a key skill as scientists we need to know is to communicate. We need to communicate. Don't be, sometimes I tell myself, especially when I was younger in the field, you know, don't be afraid to ask questions in a seminar, in a meeting, uh, not to be afraid that to say, I don't know, or I'm not familiar with this. These are things that, for me, they're kind of icebreakers, and these are soft skills that I learn. I mean, this is a learning curve. For me, this is what worked. And at the end, we need to communicate in our work, whether it's to communicate our results, whether it's to communicate via a lecture, via poster to students, to colleagues, to peers, to our group, to our unit. You know, it's all about communication, how to do it. Sometimes we give lectures to the lay audience and we have to tailor everything. So this is something very, very important that I think we need to work on. Um, yeah, during a career. Yeah.
Yeah, I also made the experience that communication really is key. Being able to talk to different people inside or outside of your field, that's super important.
Not only to talk, but also to engage them, you know, to raise their to scratch the surface and raise their curiosity and interest. And if I feel that I left and they're going home with these questions and still wondering about, oh, that was interesting, that was nice, that I learned about this and that, I think that's nice. I like it when it happens to me, when someone does this to me.
Yes, absolutely. And do you have any tips or tricks how to engage people?
Just be natural, I think. Just be natural. I remember that I actually learned from a friend of mine, and she made it a point that, and we were still students, she made it a point that it's like a challenge. Every seminar or every lecture, she must ask a question. She must find a question to ask. And I said, wow, okay, that's a challenge that I would like to take myself. And I took it. And yeah, and there's no silly question. Maybe I couldn't understand. Maybe the information wasn't conveyed. But I mean, we're about communicating, and I find that people always like to explain about their own work. And so do I. Okay, I can be frank about it. So if it shows interest and curiosity, I think it's worth it.
I was wondering, looking at the current technologies and trends in science, like imaging, omics, and computational tools, how have you adapted to these?
Well, that's a challenge. Omics, I don't think I've adopted yet. But I did transition through different methods and concepts as my science evolved, as the questions that and the systems that we had to use changed according to the scientific question. For instance, we study cancer, and suddenly I had to find out all about cancer models and in the mice and in vitro and how to do, we learn everything. I learned it with my students, which was kind of nice. And it also contributed to the feeling that I'm also personally growing in the field and that I'm learning myself, and that's quite nice. And then we do some trauma models. So we ask questions about trauma, and then we have to adopt a little bit of trauma models. Immunology, this is about immunology, inflammation, and so on. Originally, I'm not an immunologist, so I'm I'm still learning to be an immunologist. I don't have the basic immunology, courses behind me, but I think that I'm getting it as I go. I learn from meetings, conferences, chats, and stuff like that. Um, but today I feel that it's a strength for me because my toolbox is full of many, many different diverse technologies. I feel secure and safe that I can ask questions in developmental biology, in neurobiology, in cancer, in inflammation, immunology. Um, yeah, I think that I'm happy to learn other things, although it is a challenge at times. And when I embark on a project where I do not feel that I'm, you know, standing still, then I prefer to do this with a collaborator that is more knowledgeable about how to do these things.
That's so true. That's so, so true. You were mentioning a lot of your students in this talk so far. What advice would you give to somebody who is just starting in the field?
Communicate, go to meetings, expose yourself, become exposed to others, um, learn about. There are many styles and models. Everyone does whatever is good for them and best for them. Personally, where they feel most comfortable, I believe that's the best way to do your science. And I think it's like any other field. So this is my advice: to get to know people, to find your comfortable place in the field, get grants, obviously, get good students and nice ideas, and go at it.
Well, let's move on to something more uh, life, daily life. How do you balance your personal life with your scientific career?
First, I want to say, depends who you ask. My husband would say that I work too much, but and he's also a scientist. But I think I'm good with a life work balance. I do things because I want to do them and I enjoy them, and I feel that I am committed to what I'm doing. If I stay up late until, I don't know, sometimes 4 a.m. in the morning, it's because it's an internal drive and I feel that I want to do it, and it gives me fulfillment at the end. Life work balance, life. So maybe a little bit about myself. I mean, when I was a PhD student, I had my two first kids, and then my first postdoc, I had my third kid. And then we moved to my second postdoc. It was my husband's first postdoc, to the US. 2 postdocs with 3 kids. That was certainly something unusual in the scene. But yeah, today they're grown up, and I'm proud of all of, the family. So I think I've done well. I don't think that people should postpone any of their life for work or work for life if possible. It's easy to say when I was able to make my own decisions. Sometimes, yeah, okay, for life, it might have been better if before I came back to Israel to start my own lab, I would have stayed another year as a postdoc because I would accomplish much more. And but my family, our kids, they had their own needs, and I just weigh the pros and the cons here and there, and I believe that at the end things can work out. If we make them work out, things can work out.
That's so inspirational. I'm glad that's so inspirational for us as female scientists. I think that's such an inspiration. You were talking about your postdoc that you had sort of to give up for moving back again to Israel with your kids. Apart from this, do you feel comfortable of sharing with us any other sacrifice you have done uh, from your career, and if it was worth it?
I don't think really that I made any sacrifice. I mean, really, I think that everything I did was out of choice. And it's kind of by default, you do one thing, then y- you don't do the other one, or you sacrifice if you want the other one. I don't see it as a sacrifice, and I believe in flexibility and adjustment. So if you go somewhere and it's not perfectly what you want, you can make other things happen.
Moving on with the future. What challenges or opportunities do you see for the next generation of scientists?
Well, today we live in a very turbulent world, I would say. Politics, funding, politics become more and more involved in affecting science. I think that's quite an issue, especially in the US. I think AI, fake information. I think these are challenges that we're learning how to deal with today. You can find fake figures, AI generated figures, not to mention you can see sometimes in papers these AI generated texts. And I think this is something that we need to learn how to deal with. That's going to be a challenge. And the politics, obviously. Things are becoming, the world is and science is becoming too political. I think it has to be kind of wrapped, in cotton balls and very, very protected, because science is neutral. Science is for everyone.
That's so true. And if you could go back and speak to your early career self, what would you say to her?
Well, I think I would do everything the same way. I would just rewind and play again. I think that I've been told when I was a young scientist, my PhD years, both first postdoc, people, my PI, my mentors, they said, you have to enjoy, you have to really love to come to the lab because you spend most of your hours there, and you have to like what you're doing. So I liked it. I really, really liked it. And when we have this basic curiosity, every topic that you just start to scratch the surface of, everything is fascinating. Everything, you can find questions and stuff to explore everywhere.
What challenges do you believe the field will need to overcome in the next years?
Well, I think, especially in immunology, and this is something that I feel in meetings. There's so much knowledge gained in immunology. And one of the and it's being translated. Obviously, we want to translate what we know for the benefit of mankind. One of the challenges, I think, is that still very little people benefit from what we gain. For instance, immunotherapy. Billions and billions of dollars have been invested in immunotherapy, CAR T, but how many people really benefit from this? It's just a very, very, very thin layer of the cream of these people in the world. And I think one of the challenges would be to implement our knowledge so that the mankind around the globe could benefit from this.
Coming up to our last question, could you share with us some a memorable anecdote that you have in your career, perhaps something amazing, unexpected, or even surprising that you will feel comfortable sharing with us?
I'm thinking about two things. One was they have to do with the same project. So when I had to generate a conditional knockout mouse to study the role of protein S, I generate this conditional knockout, and I generated ES clones. Of course, you have to do the cloning. You have to replace the gene or insert LOXP sites, the 5 prime, the 3 prime LOXP. And you have the clone, the positive clone, which is then injected into the mice. So I isolated this clone. You grow all the clones in these 96 well plates, and the chosen clone, I had one clone. I gave it to the transgenics department, and something happened. They couldn't grow this clone. The protocol said after I give, or after I transfer the clone to the transgenic department, there's no use of this 96 well plate because it already thawed and it has to be frozen. But I like to be on the safe side, and I don't know, something got into me. I just wrapped it again in parafilm and whatever I had to wrap it in, and I chucked it back in the -80. And then when I got this news that they didn't manage to grow the clone, I said, wow, wow, wow. This was like very, I've been working for a year and a half on this, and now I have to do it, well, not from scratch, but it took me back. It would have taken me back a few good months. And it was kind of the time that I didn't know really what to do. Maybe it was a weekend. We went to a Chinese restaurant, and the fortune cookie had this little thing, the fortune, which said something that I interpreted like, if you have a tough time, you will see that it will work out. I didn't really know, it's a very banal saying. Everyone, you can adopt it to anything in life. But I decided, okay, I kept this little note from the Chinese cookie, the fortune cookie. I put it in my notebook, and I still have it till today. And I went back to this 96 well plate. Everyone in the lab said, no, don't even bother. There's nothing live there. I scraped everything I could. I gathered every microliter, anything from there, and I managed to grow one colony. So there was one cell that grew to one colony, and this one colony grew, and this is the colony that I gave to the transgenics department, and these are the mice that we work on more than 10 years after. So yeah, that was a roller coaster, but eventually, yeah. So that was one anecdote. Another one that I remember is that at the end, so the homozygous mice are embryonic lethal, and I didn't get any homozygous mice. Of course, we get heads first, and then homozygous. And I wanted to see at what phase they die. So at a certain point, I took the pregnant downs and I opened them and I went to the stereoscope and, I opened the uterus and the embryos pop up. Whoever has done that, they know that the embryos just pop up if you, uh, poke them with a forceps. And the first one was normal. The second one was normal. The third one was all full of hemorrhages and it was dead. It was like growth retarded. It was dead for maybe a day or two. And that moment, I said, wow, I'm the only one in the world sitting here in this tiny, in the lab, in a bench somewhere in the world. Zoom in. It was at the Salk Institute. So the USA, zoom in San Diego, zoom in the Salk Institute, zoom in the lab, the stereoscope. And here I am. And I'm the only one in the world that I see this. And I know this. And this knowledge now is mine. And this was it was kind of a moment. It was a wow moment for me. I'm the only one in the world who knows this. And it also, at that moment, I said, wow, it's so powerful what we're doing in the lab, because up till now, I was just doing the constructs and making sure. And all I was interested in was bands. Oh, I got the band at the right molecular weight. And this restriction enzyme cut the band. And I was, happy to see bands. Though that's what I got excited about, because my progress, it showed progress. And then suddenly from this DNA, it becomes an organism. It becomes an animal. And it's detrimental. It's embryonic lethal if I knock it out. And I can see it.
What
And it was a very, very strong moment for me. I remember that. Yeah. And I wish everyone, I really wish everyone in science can have one of those moments that they've worked on something for a while and they know, and then they enjoy for a few moments this wow moment that they realize what they're doing and it has consequences.
Thank you very much for all your time. And thank you very much for all your experiences that you shared with us, all the knowledge you shared with us. We finished this session with a lot of learnings after what all you shared with us. So thank you very much for your time. And we really hope to keep on reading about your research.
Thank you so much, Tali. It was really inspiring to hear your journey and all your advice and tips and tricks.
Thank you very much. It was a pleasure for me, really. Thanks a lot.
That's a wrap for today's episode of Climbing the Immune Ladder: Steps for Success in Science, iiSIAR podcast. A huge thank you to Tal Burstyn-Cohen for sharing her insight stories and experiences with us. We hope you found this conversation as inspiring and thought-provoking as we did. If you enjoyed the episode, don't forget to join us on the next episode and share it with anyone who might benefit from it. You can also follow us on our webpage to stay updated on future episodes and innate immunity updates. Thank you for listening, and remember, persistence and asking the right questions are what drive science, and maybe even your own journey. Until next time, stay curious.